Hi everyone, welcome to 𝗖𝗵𝗆 Sutra. After the introductory lecture, this is the GIT infection class and this is the first class and we are going to discuss about the food poisoning topic. And in food poisoning topic, the organism of discussion is going to be Staph aureus. This is Dr. 𝗵𝗺 and I am your microbiology faculty and mentor. Guys, let us understand Staph aureus because this is the most important organism, because all of you who are practicing doctors, all of you who are going to your clinics, Staph aureus is the most common organism which is there on your hands. Let's understand the classification of Staph. See, you all know that Staph is a gram-positive cocci. If we read about the classification, first of all, please understand that either the Staph is coagulase positive or Staph is coagulase negative. If you want to know about coagulase positive, this is Staph aureus, and the one which is coagulase negative has its name as coagulase negative Staph. In coagulase negative Staph, we check for novobiosine. Novobiosine. The coagulase negative Staph, which is novobiosine sensitive, is called as Staph epidermidis, whereas the one which is novobiosine resistant is Staph saprophyticus. Okay, so let us understand a little about Staph epidermidis and saprophyticus, but please remember that these both are not important if you are reading Staph for GI. Okay. Staph epidermidis, remember, it is a skin commensal. It is there in my skin, your skin, everybody's skin. The function of it is to form a slime layer, biofilm formation. It forms a biofilm, okay? A biofilm is formed by them. But remember, if it goes inside, if Staph epidermidis goes inside and the organism is very fond of, this Staph epidermidis is very fond of prosthetic valve, my dear friends, very fond of prosthetic valve, they can cause prosthetic valve endocarditis. This I have already told you when we were discussing the infective endocarditis. Okay. So these 3 points. First of all, it is a skin commensal, forms the biofilm. If got inside, it is responsible for causing prosthetic valve endocarditis. Second is novobiosine resistant, that is Staph saprophyticus. My dear friends, Staph saprophyticus is responsible for causing UTI in the young women. That is why it is also called as honeymoon cystitis, because it is going to happen in the young women, called as honeymoon cystitis. So honeymoon cystitis is caused by Staph saprophyticus. Now Staph is very important, but how will you diagnose that it is Staph? So remember, Staph is gram-positive cocci in clusters, grapes. I always say Staphylo. Staphylo, philo word we use for love, and everyone should love grapes. In fact, I love grapes. So Staphylo wants to live as a grapes. Okay, in grapes. If you talk about mannitol salt agar, the Staph aureus ferments the mannitol, because mannitol itself is sugar and it is fermenting, and when it is fermenting, it leads to golden yellow color colonies. This golden yellow color is similar to oil paint. It's just like the walls are painted with oil. So Staph aureus fermenting the mannitol and producing yellow color and oil paint colonies. But remember, Staph aureus is a common cell in the anterior nares. So Staph aureus, A for aureus, A for anterior nares. Okay. Virulence factors. So please remember, virulence factor, first virulence factor is exotoxin. Now exotoxin means what? See here, you have got a T cell. T cell has got a receptor and you have got alpha chain and beta chain of these receptor. So the super antigens are the antigens which bind to the beta chain of the receptor. So that is why the exotoxins, if you remember our lecture from general microbiology, if you have not seen, please have a look at that. I have told you that exotoxins of Staph are the super antigen, means they are going to bind to the beta chain of T cell receptor. Now, what are the example of exotoxin? The first example is exfoliative toxin. This exfoliative toxin is responsible for causing staphylococcal scalded skin syndrome. So, staphylococcal scalded skin syndrome is caused by exfoliative toxin, which is a type of exotoxin. The another exotoxin which I have talked in general microbiology is TSST1, toxic shock syndrome toxin. So, remember like this, P, Q, R, S and T. So, T for toxic shock syndrome toxin, S for staphylococcal scalded skin syndrome. Now, this toxic shock syndrome toxin is associated with nasal and vaginal tampons. And what are the symptoms? Obviously, because it is a toxic one. So, toxic one means it will have infection, and infection leads to fever. Then because it is a shock, so it will have hypotension with it, right? And because less blood will reach to the brain, it can lead to confusion. Because it is having toxin, so toxins can cause vomiting, can cause diarrhea, and a typical rash which is called as sunburn rash. So, S for sunburn and T for toxic shock syndrome toxin. So, please remember this, this is very important. So, in the virulence factor, first of all we discuss exfoliative toxin, then we discuss toxic shock syndrome toxin. Now, the next one is the enterotoxin, the very important one responsible for causing the food poisoning. This is really important. Remember enterotoxin, enteron means gut. So, entero means gut. So, these are gut toxin. My dear friends, please remember S for staph, S for stable, okay, and S for sooner. That means the toxin, the enterotoxin caused, formed by the Staph aureus is heat stable. That's why I told you stable. Sooner, actually it is a pre-formed toxin. Already Staph prepares it. So, it is pre-formed. So, it is going to cause infection very soon. It is going to cause infection very soon. And then next S is for salad. So, these are found in salads. Okay. The other things are custards, ice creams. Ice creams are actually very important. Please don't forget them. And because I told you that these are pre-formed, the incubation period is of only 1 to 6 hours. So, if after 1 to 6 hours of eating ice cream or the custard or the salad, you are developing vomiting, diarrhea, then you can suspect Staphylococcus aureus. But please remember, S is also related to second. Now, what do you mean by second? Second means 2. So, it is going to do 2 things. What are the 2 things? Vomiting and diarrhea. But can you see vomiting has 2 sticks? So, remember vomiting comes first, then the diarrhea. Second 2 things having 2 sticks. So, vomiting, then diarrhea. This was the first enterotoxin. So, I am teaching you what? I am teaching you exotoxins. One was exfoliative toxin. The other was toxic shock syndrome toxin. The third one is enterotoxin. Coming on to, after the exotoxins, coming on to protein A. So, protein A is something that binds to Fc portion of IgG. So, this is the structure of IgG. Okay. So, you have got a variable portion and a constant portion, which is called as Fc, and this is a variable portion. So, this is Fv. Now, protein A is very smart. See, if anything binds on Fv, it is going to get killed. But if anything binds on Fc, it is not going to get killed. So, protein A is very smart. It binds to the Fc portion of IgG, which prevents the phagocytosis, Coming on to the enzymes. What are the different enzymes being produced by them? So, they produce DNAs, which is going to kill the DNA. They are catalase positive. They are hyaluronidase positive. They are phosphatase positive. They are tellurite reductase positive. Okay. So, I am a big fan. You must be a big fan of ChatGPT. So, ChatGPT. Okay. So ChatGPT means P for phosphate, T for telluride. So see P, T, then H for hyaluronidase, C for catalase. Okay. So remember, this is a mnemonic which you can remember easily, and one more is obviously the DNAs which you have to keep in mind. Okay. So ChatGPT's initial 2 and last 2 letters can help you remember the name of the enzymes. Okay. And yes, initially only I told you about the coagulase. Coagulase is important, right? So coagulase positive, how to check? So remember, this coagulase can be a free coagulase or a coagulase can be bound coagulase. In order to check the free coagulase, we do the tube test. So T and F look similar. So tube test is for the free coagulase, whereas slide test is for the bound coagulase, and this slide test is for the bound. So it depends upon the bound, that means clumping factor. Okay. Next important part is the cytosine. The other part which is released is the cytosine. Remember, the various cytosines are beta hemolysin, alpha hemolysin and gamma hemolysin, and PVL, pentan valentine leukocidin. Okay, these are the substances released by Staph. One is the beta hemolysin. Beta hemolysin, remember, Staph aureus produces hot and cold phenomena. That means at 37 degree where there is hot, it is going to undergo lysis, beta hemolysis. It undergoes lysis, but it is not visible at 37 degree, it is visible at 4 degrees Celsius. So at 37 degree, it will go lysis, but at 4 degree, it will be visible. This is called as hot cold phenomena. Also, it has got alpha and gamma hemolysis, and please remember, Staph aureus releases valentine, pentan valentine leukocidin, that is going to kill the leukocytes. Can you see leukocidin? Leuko means white blood cells and cidin means killing. So pentan valentine is going to kill the WBCs, the leukocytes. This is the pathogenesis. Coming on to the clinical feature. So the mnemonic is soft pains, my dear. S for staphylococcal scalded skin syndrome. O for osteomyelitis. So the most common cause of osteomyelitis is this only. F for food poisoning. T for toxic shock syndrome. P for pneumonia. Okay. So soft pains. I for infective endocarditis. I told you the most common cause of native wall infective endocarditis, especially in IV drug abusers. In IV drug abusers with native wall infective endocarditis is this only. N for necrotizing fasciitis. S for surgical site infection because your hands are full with Staph aureus. So this is the mnemonic soft pains. Coming on to the treatment, my dear friends, remember, penicillin used to be the initial treatment, but Staph aureus started producing beta-lactamase. So penicillin is the beta-lactam and Staph aureus started producing beta-lactamase. So now this is not able to act on Staph aureus. Penicillin is a beta-lactam, no, and Staph releases beta-lactamase. So they kill this penicillin and penicillins can't work. Now you will tell me, where are beta-lactamase kept? In bacteria, in the comment section. Then we started giving methicillin. So what happened? Methicillin also, bacteria did what? Bacteria started producing a gene which is called as MBBS gene. Now this MBBS gene was responsible to kill methicillin. So you give methicillin, bacteria has MBBS gene, MBBS gene will destroy methicillin. So bacteria became MRSA, methicillin resistant Staph aureus. So what you did? You started giving vancomycin. But bacteria is still very smart. Bacteria started producing resistance to vancomycin by VAN-A gene. So now new bacteria, VRSA, vancomycin resistant Staph aureus. Then you started giving linezolid, daptomycin, streptogrammin and mupirocin. So ultimately, in order to kill Staph, these are the things which we are going to use nowadays. But my dear friends, please keep in mind, If your patient is suffering from toxic shock syndrome, you are going to give clindamycin. Clindamycin is going to cut the toxin. CUT, cut, TOXIN, so TT, okay. So clinda is cutting the toxin, it will cut the production of toxin. And if your patient develops food poisoning and you suspect that the food poisoning is because of staph, no antibiotics should be given because it is toxic mediated, toxin mediated. And what to do? Please treat the patient symptomatically. Okay, if there is fever, you give paracetamol. If you are having the pain in stomach, give 𝗵𝗲𝗵𝗺. Okay, give ORS, give a lot of fluid. So this is the treatment. So laboratory diagnosis we have discussed already. Okay, the stain, the morphology of the stain, you will see in light microscopy. The culture medium also you have checked. The culture morphology, that is the oil paint colonies, you have already written. Okay. Now specimen, which specimen you will collect? Either the pus specimen or the fluid or the tissue specimen or the sputum specimen you will take, and this is the picture you are going to visualize. See here, these are present in clusters, these are present in grape-like manner. Okay. So this is all about the Staphylococcus. We have discussed staph, aureus we have discussed, epidermidis, saprophyticus we have discussed. Thank you so much for watching. We'll see you in the next class.